Hi everyone. In this video, we'll talk about the polymerase chain reactions, the real-time polymerase chain reactions, the reverse transcriptase polymerase chain reactions, but RT-PCR actually stands for the reverse transcriptase polymerase chain reaction. We'll talk about the interpretations, especially in the terms of COVID-19 infection. How should you do it? But the basics fact as to the mechanics of the PCR, the real-time PCR, reverse transcriptase PCR, and more importantly, what do you interpret and how do you interpret a PCR report? Let's start with it. The first thing you should understand here is the structure of the COVID-19. A structure of the COVID-19, it usually has this structural genes and it has a non-structural genes. See, a non-structural genes would be something which is inside the virus and is helping it either for an enzymatic purpose. For example, in this case, it would be the reverse transcriptase. So, reverse transcriptase is RNA dependent. RNA dependent polymerase. So because it is actually depending on the RNA, because the virus has an RNA, it can make a DNA from the RNA. It can make an RNA from the RNA. So that is how you call it the polymerase, RNA polymerase or a DNA polymerase. So this is actually having a RD-RP gene. So RD is RNA dependent. RP matlab RNA polymerase. So RD-RP gene. But that is actually inside the virus. What is on the structure here? As you see this virus, they have the heme adenine esterase. They have the membrane protein. So M gene will make the M gene will make the M protein. Will have the envelope protein. So E gene will make the envelope protein. Now then there is a nucleocapsid protein. Okay, nucleocapsid protein again is a capsid and hence it's not a non-structural, it's a structural gene. So N gene will make the nucleocapsid and the RNA, and the spike S gene will make the spike glycoprotein. So we have a 4 type of genes here. Again, that should be very clear in your mind. It has a S gene, it has a N gene, it has a E gene, it has a M gene. But what about the RNA actually? So RNA is made by something called as RD-RP gene, which is then made by some open reading frames, ORF gene. Let's talk about those also. See, what happens is, if you look at the virus structure again, so it has basically some ORF A and ORF B. So majority, I'll say 2/3 of the entire genome of this COVID-19 is actually composed of the ORF, open reading frames, 1A and 1B. Okay, now because they don't code for a structure, we call this non-structural proteins. Now, look at this, 1/3 of this entire genome is composed of the S gene, E gene, M gene, and N gene. Remember this basics because this will be very handy while you're interpreting a RT-PCR report. We are interpreting a RT-PCR report. Okay. Now, before we now move forward, let's now understand what is exactly a PCR. Okay. What is PCR? Before we move to RT-PCR, let's make our basic correct. What is a polymerase chain reaction? Remember, this was discovered by Carey and Mullis in the 1993, for which there was actually given the Nobel Prize, and I think that is the well-deserved prize, isn't it? Now, what they did was they saw that a DNA is actually a double-stranded DNA, and with the help of some temperatures, you can actually separate the strands of the DNA. Okay. Then what they did, they tried to increase the number of the DNA by adding some DNTPs. What is DNTP? They actually are the nucleotide triphosphate, which can actually add the A, T, C, or G according to the corresponding purines or pyrimidines. For example, as you all know, A combines with T and C combines with G. So let's see what they did. The first step they did was a denaturation. What they did was, when they raised the temperature of the PCR to actually 95 degrees Celsius, what happened was, because of this entire situation, what happened was, so what they saw was, at 95 degrees Celsius, at this temperature, a unique thing happened. At this temperature, they found that when they're putting this, the H bonds, you remember the H bonds between any A or T, if there is A and T, if suppose there is A and T, I will draw it here only, you will understand this better. Suppose here it is A, T, G, C. So what is corresponding here? Here it is T, it is A, it is C, and it is G. Now understand the triple bonds and double bonds. A and T has double bond, A and T has double bond, well C and G has triple bonds. Now these all bonds are actually what? These are hydrogen bonds. So at 95 degrees Celsius, there was breakage of the hydrogen bond. That means this bond that was actually putting all them up together, they actually were lost. in the 900 degrees Celsius. So the two strands got separated out. Then what they did, they actually brought the temperature to 55 degrees Celsius. At 50 degrees Celsius, it was called as annealing. At that temperature, what they did, they added primers. Now, what is primer? It is exactly a short segment of DNA with complementary base pair to the original DNA. That means, let's see, if suppose there is G, C, C, T, and A at the end of this strand, let's assume it's 5-3- and it's 5-3- on this side. So it's 5-3- and there's to 5-. So if I want to make a primer, I'll just make a complementary strands. For example, C will compare with G, C will compare to G, C will combine with G, T will combine with A, and A will combine with T. So if I actually make these all nucleides to come together in my laboratory, so I can make basically a primer which can attach to this part of the DNA. Similarly, they also added one part of primer here. So you can call this as forward primer, you can call this as forward primer, and you may call this one as reverse primer. So forward and reverse primers were added at a temperature which was actually 55 degrees Celsius, and because it was annealing here, this was annealing, it was called as annealing at this step. Then what it did, they have did, they had separated and they have annealed. What is the next thing? You are expecting this primer to make the DNA here and this primer to make the DNA here. But what you need for that? You need an enzyme called as DNA polymerase. So what they did, they got this DNA polymerase from a thermus aquaticus, which actually source of this DNA polymerase, and because it is actually stable at high temperature, and therefore it was basically used. So at a temperature of 70 degrees Celsius, this temperature you should remember, they added DNTPs and catalyst of magnesium chloride. So what they did when they added this, so you have every masala here. You have basically the basic DNA of interest. You have the primers you have already put, forward and reverse primers. You're adding the DNTPs and magnesium as a catalyst. So they keep on making on this side and this side with the help of this unique enzyme called as DNA polymerase, in this case called as TAC polymerase, because it was actually got from the thermus aquaticus weeds. Okay, this is basically what they did. Now, if I want to make you understand in a very beautiful manner, let's see some animations out of here. In this entire video, I'll talk about how it actually happened. See, to make this happen, you have to understand the basic principles of a PCR. So what happens in this PCR actually is, I'll talk here with the reaction mixture. A reaction mixture will consist of all the things you want to. You have DNTPs, you have the RNA polymerase and the RNA primers along with the RNA of interest. You program a setup, and what you do at denaturation at a high temperature, you have a 94 degrees Celsius. Okay, this is at a high temperature. So what happens? The DNA can easily separate out. Look at the DNA getting separated out here. Now what happens? The hydrogen bond actually breaks here. Okay, hydrogen bond actually it breaks here. Next one, what happens? So after the n number of cycles, what you appreciate is the number of DNA will be 2 to the power n. That means after one cycle, it is just a 2 double-stranded DNA, but after 3 cycles, it becomes 2 to the power 3, which becomes 8 double-stranded DNA. Now, this is what is about the PCR, polymerase chain reaction. Can we think about, now what has happened here is, in a PCR, what has happened is, we have tried to amplify the DNA. But the big question is, if you are amplifying a DNA, can we detect what is the amount of amplification? If we can, it will be called as quantitative PCR. But if we can detect the same thing while the reaction is happening, at the real time while the reaction is happening, it is called as real-time PCR. Understand? PCR was just an amplification, right? PCR was just an amplification here. If you're trying to detect what is amplification, you can call this quantitative PCR, or you can call this a real-time PCR if detection is occurring at the time of amplification. So how do we do that? For that, let's move to the next level. We now understand what is a RT, reverse translated PCR, or a RQ, that is real-time quantitative PCR. Okay, let's move forward with that. The entire basics lies here. What happens in this whole thing is, see, what is called Tachman probe. Tachman probe is a unique probe in which it has two areas. It has a fluorescence emitter and has a quencher. Quenching a thirst. So what happens? A fluorescence will actually emit the fluorescence light. But if the fluorescence is quite near to the quencher, that emitted light will be quenched or taken by this quencher. This type of probe is called as Tachman probe. It's called Tachman probe. Now what happens whenever the fluorescence and the quencher are quite together, quite near to each other, they will never be able to emit the fluorescence. But when this and this, they will actually separate out, there will be emission of the fluorescence. Now, what and how it was done is, let's see forward. So what they did was, as I told you, when these two are together, there's no light emission. But the moment these two are separated out, there will definitely a light emission. And the light emission, the amount of light emission would be proportional to the amount of DNA getting amplified. It is called as Tachman probe. So what it was done is, if you look at this, initially what we do when we do a reverse transcriptal PCR, what we need is, we actually need that is, we want to first make the DNA from the RNA. Now, because we want to first make a DNA from the RNA, it becomes reverse transcriptase. Because we are now making, see what we normally do is, we first take DNA and from a DNA we make RNA and the RNA makes a protein. Now, you know the basic steps. This first step is called as transcription, right? And this is called as transcription. And the next step is called as translation. What we're doing here is, we're making the complementary DNA. C means complementary. The word C refers to complementary. So we are making actually the complementary DNA from the RNA. Now this process is called as reverse transcriptase because it requires an enzyme called reverse transcriptase enzyme. Now because now we have got a DNA, we can now do a usual PCR. So the first step, this step makes it what is called as RT-PCR, the reverse transcriptase PCR. So what we take, first take a sample, and you all know the sample is either a nasopharyngeal swab and an oropharyngeal swab. Most of the laboratories take both the sample. Some of the laboratories take only one sample, but it's often better if you take both the samples the same thing. You send the sample in a viral transport media. The viral transport media eventually actually kills the virus, but at the same time makes the RNA non, or you can say, viable. The RNA is not killed there. The virus may be killed, it will not be able to replicate there, but yes, the amount of the virus will be same at the time of collection and the time of the sample entry into the lab, but the virus is only killed, not the RNA in the virus. It is called as viral transport media. Then what we do is, we put the sample with the lysis buffer. The lysis buffer will lyse the virus. I hope you remember the virus structure I showed you. There was the outer structure and the inner RNA. Unless you don't break the structure, you cannot take out the RNA out. So you need to lyse it. The lysis buffer has phenol and guanidine isothiocyanate, which you must remember here. After this, what happens? We put there a spin column. A spin column is something like this. It is something like this. In a spin column, there are some net-like structures here. In the net-like structure, what happens? Everything that you don't want, that we see, RNA will always have some proteins with it. So do you want proteins? You don't. So what we do is, we put this entire content here and the proteins will come here, while the RNA will get entangled at this area. The RNA gets entangled in this area. This would be RNA only. Okay. So RNA is actually getting extracted out by this something called a spin column. Okay. Now what we do, we need to elute out the buffer. Elution buffer means, elution we will actually elute, means separate out. Elution will separate, it will separate the pure RNA out. So what we need first of all to lyse the RNA, lysis buffer. Then you need a spin column to separate the RNA, and then you want to elute the RNA, and hence we are using this elution buffer. Now what you need a reaction mixture. A reaction mixture, you all know, will have the DNT piece, will have the primer forward and reverse, will have the DNA polymerase. So what is added and what is extra to the normal PCR? We have this one and we have this one as an extra thing you need from a normal PCR. So because I explained to you normal PCR, I don't think there will be any difficulty in understanding that what you else need. You need a Tachman probe for a actual real-time PCR and an RT enzyme for a reverse translated PCR. So basically what you need 1, 2, 3, 4 and 5 masalas to make this entire thing. So you have taken this mixture and waterizing it, that means you're mixing it and putting in a PCR machine. You're putting in a PCR machine. Let's understand and let's look how does the PCR machine actually looks like. I'm showing you a PCR machine from my own lab. It'll be very easy for you to understand that way. Dekho. Now what happens is, when we run a PCR, I told you about this whole thing. What you're seeing here is, look at this RT-PCR machine. RT-PCR machine, it has some fluorescent light emitters of various wavelengths. And look at this area, this is where you put the samples. So usually our RT-PCR machine has 96 wells. So this RT-PCR has 96 into 2, so you can actually do 96 into 2 tests at a time. Even though you have to put some controls, positive and negative controls, to ensure that you sample how do you look at it, okay? But at least they will have 96 wells. So this PCR machine has 96 into 2. That means it can actually do 96 into 2 tests at a time. I will take 2 positive controls and 2 negative controls. You have to subtract that also with the same machine. Now what happens? I told you about, right? I told you first of all also about this Tachman probe. The Tachman probe basically it has reporter and a quencher. In this case, the fluorescence is getting set as R. So reporter and quencher, when they're together, there's no emission of the light. But once the reporter and quencher they get separated, they will always emit light. Now, how will they separate out? I'll talk about this in my next part of the video. Okay, see. So what happens here is we have put entire thing in a reaction mixture. We have put the entire thing in a reaction mixture. What next? See, the gene amplifier guide or that you can do is you can look for the RDRP. What is RDRP? It is the same as this OPA reading frames, ORF, 1A and 1B. You can look for the E gene, you can look for the N gene, you can look for the S gene, and you can look for the M gene. But you will see most of the laboratory reports, I shall also show you, they rely mostly on the ORF, E gene and the N gene. Okay, E gene and the N gene. So what and how does this reverse transcript PCR work? Now see, this is a normal DNA. Okay, let's say this is the complementary DNA. Complementary DNA, that means there was an RNA. First of all, there was an RNA, and from the RNA we have made the complementary DNA. How? By reverse transcriptase enzyme, RT enzyme. Okay, this complementary DNA, it can again be used to make the other part of DNA. And how do you make it? You do it by PCR. So what I do is I'll actually first try to make the DNA to increase in the number. To increase in number, what I do is this is a complementary DNA. It's complementary DNA here, and I will have one more DNA which is actually complementary to it. So this is a 3' to 5', and this one is 5' to 3'. So how does happen? See, if you have this DNA, so what to do? You put a primer here. The primer will make every DNA here because you have the DNTPs. You have the DNTPs, so it keep on adding the nucleides. For example, if there is, let's make it like this. If there was supposedly A, T, G, C, C, the DNTPs will keep on adding itself, which is complementary to this side. For example, for A, as I told you, it will be T. For T, it will be A. For G, it will be C. For C, it will be G. And for C, it will be G. And they're all coming from what? They're all coming from this DNTPs. So DNTPs actually are what? They're A, T, C, and G. They're A, T, C, and G, which it keeps on adding as the reaction is progressing forward. Okay. Next, what happens? You have to anneal them. So for the purpose of annealing, what you do? You put temperature at 50 degrees Celsius. Now what you do? You put a forward primer and a reverse primer. For that, what you need to do? You first need to again denature it. As you know, I remember it is done at high temperature or 92-95 degrees Celsius. While in denatured, the two strands will separate out. After separating out, you put the forward primer, you put the reverse primer. Forward primer, I'm writing this P and this is P. These both are primers. And along with this, you also put a quencher or the Tachman probe. What is this? It's a Tachman probe. It's a Tachman probe. A Tachman probe, I'm repeating this again and again, that this is a fluorescence emitter and this one is a quencher. Some books will call this fluorescence emitter as R, that's reporter. Same thing. Either you can call this fluorescence emitter or you can say reporter of the fluorescence. Same thing, okay? What you do, then you have already added DNA polymerase. So what happens now? With the help of DNA polymerase, which is acting on the primer, the DNA polymerase will keep on adding the NTPs here. So the strand will keep on making here. Similarly, the reverse primer which I put here, it also takes with the help of the DNA polymerase and makes the entire strand here. But what happens when the DNA polymerase it comes and meets this Tachman probe? This DNA polymerase can cleave this Tachman. Why? Because the Tachman probe also has a sequence of 3' to 5'. And this typical DNA polymerase has actually a 5' nuclease action. Matlab, see what happens when this, let's assume this is a DNA polymerase. The DNA polymerase is making the strands here. It's making the opposite strands here. When it has reached at this point, this DNA polymerase, it will break the entire Tachman to this part here, as it will break it here. Let's break it here. As it will break it here. The moment this breaks, why it breaks? Because of the 5' nuclease action of the DNA polymerase. It breaks the probe. When it breaks the probe, the fluorescence starts getting emitted out from this. It starts emitting out from this. It emits fluorescence. Now you would say, sir, what is the significance of the fluorescence? The fluorescence got emitted only because this primer could bind to the specific region of the DNA of the virus, and therefore the amplification started happening. Suppose the original patient, suppose did not have this virus, so patient did not have the RNA. If patient did not have RNA, it will not make the complementary DNA. If the patient did not make complementary DNA because of RT enzyme, the whole reaction will not at all progress. In that case, the fluorescence will not increase. Now look at the opposite thing. Suppose the patient has RNA and suppose the patient has less copy number of RNA. So less copy number of RNA will make less copy number of amplifications. So ultimately, the fluorescence emitted by this fluorescence here would be less. But suppose a patient has huge number of viral RNA, the number of cycles required to amplify it would be less. To the point, the number of cycle matlab, let it be very clear to you. Suppose the two people, person A and person B. Now in person A, the viral load is less. Because viral load is less, so I need to amplify this. That means I need to repeat the cycle more and more to get a threshold number of the viral DNA. But suppose a patient B has high viral load, so the number of the cycles I will need is less because he already has high viral load. The number of cycles required is less to reach a particular fluorescence level. Now, why I'm talking about a particular fluorescence level? I'm telling you, this reporter will always keep on emitting the fluorescence, and quencher will keep on taking the fluorescence. That means some amount of fluorescence will always be there in a negative sample also. So unless we don't get a typical threshold limit, we cannot say the sample to be positive. Okay? So what we do is we take the number of cycles and take the fluorescence and then we plot a graph. Let me show you a graph once. It will help you to understand it better. This is how the entire graph looks like. Now in this graph, you look at this in graph. What you see in this graph is there is on this axis there is number of cycles, and on this axis here you can see the fluorescence emitted. Now see, you see this dotted line? This dotted line is threshold limit. Now see, suppose a patient has this threshold limit crossing at 21 cycles. So I will say CT value is 21 because at 21 cycle the threshold of the fluorescence increased. But suppose a patient had increased the threshold at just a 10th cycle. Only here it got increased. That means the virus load was quite high. So even at 10th number of cycle, it increased above the fluorescence. That means in the patient who has less CT value, the viral load is higher compared to a person who has a CT value, in this case supposedly of 21, has a lower viral load compared to a person who has a 10 CT value. So I'll just tell you a simple fact. It is opposite. Low CT value, high viral load. High CT value, low viral load. Now, in a simple way, this was a quite interpretation. I quite find it very useful in a newspaper article. So, RT-PCR, you have to look at the CT value. A CT value indicates, look at the number of cycles needed in the RT-PCR to amplify the viral RNA so that it can reach a detectable level. A low CT value of 29 or below is a sign of high viral load, and CT value of 30-37 is a moderate amount of viral load. But please understand, every lab will have its own value. So you cannot look at this one. It just is a layman, explanation for the things. Now you should not believe on this thing. You should believe on the facts given by that laboratories. For example, if you look at many of the laboratories, the value of less than 25 is still higher. 25 to 35 is moderate, and above 35 is quite a low viral load. But does it have a huge amplification? Yes. Initially, look at a CT value. A high viral load means the patient may get more and more infectious, and he may have more and more severity of the disease. Okay. So you have understood what is a CT value. You have understood what is a Tachman probe. You have understood what is the basic of a real-time PCR above a reverse transcriptase PCR. So what happens? See, the moment the quencher is now actually broken down, the fluorescence start emitting the fluorescence which is not quenched by this quencher. So fluorescence keeps on coming out, and amount of fluorescence is directly proportional. The amount of proportional is directly proportional to the amount of DNA in the sample. These all things are done by this typical machine called as RT-PCR machine. It's called done by this machine called as RT-PCR machine. So are you ready? Are you ready to now look for a report? Let's see if you're ready or not. Look at this report. In this report, they're looking for the CT value in the E gene and the ORF1A and B. ORF1 and A, B and B is the RDRP gene. Okay. Now see, CT value. Don't confuse CT value with the CT scoring. CT scoring is done in the CT scan. CT value is just it is a threshold value. It is cycle threshold value. CT means cycle threshold value. Now look at the CT value here. It is 25.67 of which gene? Of the E gene. And for the ORF, it is 24.88 G. So as a clinician, you should be able to understand, okay, it's just less than 25, I'll take it as a high viral load. Okay, let me show you one more report. In this report, you're looking at this again, COVID-19, look at the N gene has been amplified and the ORF1A and 1B has been amplified. There's no strict rules as to which gene should you amplify, but most of the labs look for the ORF RDRP gene. Along with that, they take one structural gene. In this case, they have taken the N gene. Again, repeat, they take one RDRP gene, which is ORF1A and 1B. Along with that, they also take this N gene also. They also take N gene also. Now, this value is 14. Now, this will be quite very high viral load. It has a very high viral load. So, usually this patient may go to some amount of seriousness, but I'm very sure he will also come out fine and more healthy in the days to come. So now you understand how to actually look for the scoring system, to how to interpret the real time and the reverse transcriptase PCR. I hope you liked the video. It was quite a crisp video to make you understand everything you need to know about the PCR, polymerase chain reaction, what is a real time PCR and what is a RT-PCR. Remember, RT-PCR stands for the reverse transcriptase PCR. If you like the video, please let me know. I can bring you many such videos for you, but only if you study well and give your valuable feedbacks to us back. Thank you. God bless you all. Stay blessed, stay healthy, and stay safe. Bye-bye.